So I want to talk today a little bit about the retina itself. So we have seen that in a couple of slides today. So my work is basically focused on the retina, basically where all the light is transduced into neuronal signals. And my interest itself is global retinal function. And global has, and I will try to highlight this today a little bit, has many meanings. And also I'm working with mice, but I'm also striving into humans now. Anyway, just quickly. So probably you know all this. This is the retina. This is our eyeball. The retina is a light sensitive tissue that lines the back of our eye. It has a couple of cell layers, and obviously we talked about rod and cones, the photoreceptors, but I want to highlight these cells here. These are the output cells of the retina. They are called retinal ganglion cells, and they sense the information, or they integrate the information from the inner and outer retina and sends this in forms of action potentials, or so-called spikes, into higher brain areas. We have many different types of ganglion cells in mice and in humans. So we obviously, for this workshop, we distinguish between image-forming ganglion cells that, for example, we have what we call a local edge detector. It's a special cell that is responding to small contrast changes. Then we have also cells that are tuned towards a preferred motion or direction or orientation. So this is all, the retina is not a camera, it's a change and feature detector, and all these different cells basically work together to encode our visual scenery for the image forming function. And then obviously we have also non-image forming function, and we heard a lot about that today. So this is data, this is now a sketch from the mouse retina. We have 6 different melanopsin or melanopsin ganglion cells, so intrinsically photosensitive ganglion cells, and they are thought to be just they are thought to regulate non-image forming functions, as we heard also today. But there's also now a recent move these days that these cells can also play a crucial role in image forming function, meaning in contrast detection and visual perception. This is all, the retina not only has many different types of ganglion cells, we have also a lot of ganglion cells. So this is, I'm talking now about the quantity. So this is an explanted mouse retina. So mouse retina is around 4 or 5 millimeters in diameter, and it has around 50,000 ganglion cells. If you zoom in a little bit, you see all these cell bodies here are ganglion cells. It's a special antibody that only labels ganglion cells. And you see the information, or how much information is potentially coming out of that retina. And this is only a mouse. So in a human, we have 1 million ganglion cells that send information every split second to the brain. And to understand, in this case, global means, what it means not only the space, the quantity of cells, but also the variety of cells, you need the right tool. We started when I was working at Newcastle University together with Professor Evelyn Saw in Senegal. We established the UK's first experimental and analytical framework for simultaneous light stimulation, structural imaging, and recording from hundreds to thousands of neurons. So I'm just saying neurons because we were not restricted to the retina itself. We were basically every cell type that just called itself neurons we could record. So the idea is that we used one of the new CMOS multi-electrode arrays. So these are arrays that, in this our case, it has a dimension of 2.7 millimeters times 2.7 millimeters, and it has 4,096 electrodes squared here. This is a high-resolution micrograph. You see one of these metal electrodes is about 20 micrometers, so the spacing is 40 micrometer. So when we then put a retina on top of that, a mouse retina on top of that, and we see here these little red squares, this is the sensing area. This is how much we can sample from that retina itself, or from that neuronal tissue. This is coupled, and it's not on this picture here, with a light projector that allowed us to play any kind of pattern or movie or just constant light. So, and this is also then basically integrated, or the signals, every signal is then sent to our amplifier as a biochem, because what we're doing is that we try to really on a millisecond level to unreveal what the pattern changes is doing. Can talk a lot, a video is better. So this is a retina. This is a retina I put on these arrays. The little yellow dots are representing spikes. This is a slow motion. And I had to reduce the numbers of spikes because to make it visible. So this is stimulus-driven response from the retina. And you see they all work together. All these cells, in a way, work together over a large spatial distance to give us the information, in this case, a mouse, information about what is going on. And this is what I'm interested in. I'm not interested so much in the single cell responses. I'm interested in network responses. So to do that, I need to have a clean and sober network, so a good isolated network of the same feature detectors, as I said earlier. And there are several classification methods available. So we started, or retinal classification is not a new thing, so started very early on anatomically. So we found 14 different types, and then we had more recently, or 5 years ago, Tom Barden and his crew found over 30 different ganglion cell types in the mouse retina using a functional approach. And then this is really recent work. With the advent of new sequencing techniques and machines, it was able to do single cell sequencing with ganglion cells genetically, and that revealed over 46 different kinds of ganglion cells. You see the number is constantly increasing. Just as a comparison, in the human and in the non-primate retina, we have around 17 to 20 different ganglion cell types standing now. So this could potentially also increase. However, all these classification, or the common classification methods, are based on anatomy or physiology or gene expression. So, and that is something that is particularly difficult, because when you think about melanopsin cells or the very high specialized feature encoders, you may not elicit some meaningful responses from them to sort or to isolate them and classify them. So you're probably best off to combine several methods. And that idea actually we had. So this is, as I said, I'm introducing now the work that I did with Evelyn back in the days when I was at Newcastle. And this is in collaboration with our friends and collaborators in INRIA in southern France, and Victoria was my master's student at that time. And a preprint is available. We try to publish this work now. And the idea of our classification method is that cells, ganglion cells, can share the same gene expression. For example, the well-known parvalbumin cells, calvitinin cells, they all, for example, parfum have 8 types, carotene has 10 types. So they all express that calcium binding protein. But however, there are many, many more smaller clusters of ganglion cells that share common gene expression, and this is reviewed or shown here in the Matterstack paper from Josh Seng's lab. And there you have whatever, maybe just 1 or 2 different types of ganglion cells have the same gene expression. And this is particularly interesting. Why do they have the same gene expression? Can we use it to get really nice isolated networks? And further question would be also why they have the same gene expression. Do they have something in common, or do they work in somehow together? So that's the goal, that's the aim. We want to use our big MEA recordings, and we combine that with pharmacogenetics. I haven't explained that. In a second, I'm doing that. And then also on top of that, we put cell labeling, and for reason I will tell then at that slide. So pharmacogenetics, what is that? What is that? So you bring in an artificial receptor, there's not otherwise in your body or in the mouse, and you bring it to a specific cell type. You target that cell type, and then you can activate that specific receptor with an inert drug, which would not otherwise anything else in your system. And this is, we use the technique called DREAD. So designer receptors exclusively activated by designer drugs. So these are, it's an engineered version of a muscarinic receptor, M3, human M3 muscarinic receptor to be precisely. And you apply clozapine N oxide, or short CNO, and these receptors are activated, and this leads to a depolarization in the neuron. There are also inhibitory versions available, but we use the excitatory one. And in case we wanted to bring that in the ganglion cells, that leads to more spiking, to more spike output. So this is, we decided to go for, in this project, for 2 genes. We decided that based on the, on the sparseness of the signal. So we wanted 2 genes that were not overall distributed in the ganglion cell layer, but very sparsely, maybe just 2 or 3 types, something. That was the aim. Conveniently, and I just forgot to say that these DREAD receptors are nicely tagged with the GFP, so that's why we see here these the cell labeling very dominantly. And this is the idea. The method is that we have a control recording without any substance in there, and then we add the CNO substance, and then we do the same recordings again, and we see which cells are more activated, and these are basically our SCN1A or Greek4, the genes that we choose. So there were some pitfalls with that. I'm not going into detail. That's all detailed in the preprint that we have there. What we had to combine that with our GFP information, or with structural labeling, cell labeling techniques. And I'm just shortly show you how we did that. As I said, we use this huge MEA array, and the density of these electrodes is very narrow. And as we see the same cell spiking on surrounding electrodes, basically. And what we do is we do a triangulation and interpolation, we call that, of the signal, of the spike amplitude. This is highlighted in our 2017 paper. So we can physically originate the source of the spiking. This is also used to isolate very precisely single unit responses. And then on top of that, we have the information with the DREAD, GFP, as I said. So we overlay that with microscopy images, and then we look at cells that did show an increased spiking behavior, and then we can basically very precisely isolate this cell type. This single unit belongs to this and this cluster. As I said, this is, in this case, I'm just showing examples from the Greek4 retinas. So they are containing potentially 2, 3, 4, 5 different cell types, ganglion cell subtypes. So we needed to find out. So now we have basically all Greek4 expressing ganglion cells, but now we need to do a functional clustering. So we had already the gene expression activated with CNO, cell labeling, and now we need the the functional aspect into that. And that's what we did here, is that we played a stimulus, a static stimulus that was similar, that was Tom Barden was using 5 years ago, which called the sheared stimulus. It contains, I'll show later a picture of that. It shows a couple of different contrast changes, color changes, and so on, and frequencies. But how to split up now the responses? And we said, okay, let's go with spike synchrony, because cells that belong to the same group spike in the same way. So they respond in the same way. And that's the idea. Example here, cell A and B belong to the same response cluster. And if you see, I haven't put the stimulus here, but this is a change from black to white. You see it's an on cell. It responds in similar ways. And there's a lot of similarity between all the spike trains between these two cells. In contrast, the other cells see here not. You can calculate that with a method called spike distance matrix. You can basically then use the spike distance matrix to do a hierarchical clustering, and exactly that's what we did in our 2018 paper. For the methodology, please refer to that. But then you come up with a lot of response clusters where you said, okay, these cells spike in the same way. As I said, I'm only showing here the Greek4 results. So this is from the stationary cells. So I divided into stationary and non-stationary. Non-stationary is everything that reacts prefer to motion or objects. Stationary is just if you have a global stimulus. And we found 3 clusters three response clusters in here. One is an unsustained cells, and I want to highlight that for a moment. Because we know already, because in 2018 a paper came out that says a Greek 4 cell is an unsustained with a high spontaneous rate, and we call that PIX-on. This was done by Daniel Kirschensteiner. And so we had basically a reference. And so the task was to find this cell in our classification method, and we did. So this is the PIX-on encoder. We showed that because the response duration, means the sustainedness, is quite high. Here left is response, on y-axis is a response versus what we call the bias index. 1 means on, or the light onset we preferred. -1 is an offset, is a dark offset preferred. So it's on sustained and with a huge baseline rate. Baseline is when no stimulus was presented, how many spikes were there. So we had a lot of spikes there. So this is validation. So we are pretty sure that, very sure that our method works. So we applied it to all the other lines, and unfortunately the time is not enough to show all these nice results. What I want to show is next, what's up next is we have now this network of response clusters. So this is, when you look here, these are all receptive fields from one and the same, or from, let's say, this is one type, and we have these receptive fields distributed over the entire retina on our MEA array. And what we now can do, instead of showing relative simple stimuli, we can show more complex stimuli. And then we can see how this network works together to encode this information. And this information is then further implemented in our platform that is led, this project is led by our collaborators in India and France. And if you're interested in that, it's called Macula. This is a graphical user interface retinal simulator. You can construct your own retina, you can play with the data, and obviously the name Macula is already hinting towards it, but we're not restricted to the mouse. We want to go towards human, we want to model the human retina with Macula. And also a little bit here in respect to the Melanopsin ganglion cells. As I said, they are often not much is known, because what is not known, because it's difficult to isolate, to identify these cells in a recording. And with our method, it's not restricted that we just show. It's not restricted to the two types that I just showed. So it could be easily implemented for Melanopsin ganglion cells and have a closer look how they interact with in the network together with other cells. I think that's pretty much it. I want to thank obviously Evelyn, and in a second instant Melinda, and all the collaborators over the time that worked together with me. Thank you very much.
Thanks very much, Gerrit, for a really interesting talk. If anyone has any questions, if they could put them into the Q&A. In the meantime, I was going to kick off. I had some questions about the spatial organization of these different retinal cells. Do you have any sort of idea why there is this kind of spatial arrangement of different types of retinal cells with different functions?
Yeah, that's a really good question. This is something that, I mean, 10 years ago, we thought that every retinal cell is evenly spaced, or every ganglion cell is evenly spaced over the retina. But these days, we really find out that we have hotspots of densities of cells. That makes sense if we think about ventral dorsal, about looking in the sky, looking on the ground, looking left, right. And so there some kind of, I mean, this is mouse data. Mouse data does not have a macular fovea where we have basically the sharpest point, but the trend is coming. I mean, the human retina does not stop after the fovea. We have a lot of other cells and more rod-driven cells, circuits after that. Yeah, that's a good question, and I think we as you see, if you're going from 14 to 30 to 46 types, you see how complex it is to puzzle out, or to separate a visual scene, and with what kind of encoders you need to really think about how to puzzle it in the brain back together to make a see fast and quick, and also regulate the non-image function and how they basically play into that. So I think, first of all, you need quite a lot of new methodology, and as I said also in the last part, modeling is here very important, because all these, the gaps in between our knowledge could be really solved by putting a good model into that.
Yeah, no, that's great. Has there actually been done that much sort of modeling work of you sort of hinted at your tool that you've got?
There's a lot of modeling going on in single circuit. You know, you have a couple of cells, then you have a couple of cells in the inner retina, and then you have a couple of output cells, but not on the scales of thousands of output cells. So that's what Macula is doing. It's a layer model. So we have really input is thousands of cells and thousands of cells. We want really to use that factor space, you know, that is what is going on in 300 micrometer, was going in 1 millimeter, was going on in 2 millimeters when you present a specific image there. So there must be new clues beyond that. There are new clues behind that. We already know that even if you have a stimulus that's very far apart, and something is going on the other side in the retina too, because even if they're not stimulated, but something is going on there. So this is it's interesting, and as I said, it's not a camera. It's really a very clever feature detector and change detector.
Great, thank you. We've got a question that's come in from Barath, who has said, very nice talk. I was also wondering about spatial organization. I don't know enough about the retina, but the analyses you detailed looking at co-expression of markers or temporal firing synchrony to find clusters did not ever use spatial information of spiking units to cluster. Is there a reason for that?
It is. So I hope I understand that. So we use, with the co-expression, the markers, the cell labeling, then we have the firing synchrony, and now we want to bring in the spatial information of spiking. The spatial information of spiking is indirectly used. As I said, every spike we can sense. We can basically interpolate it out, follow it back to the origin. So this is used to do what we call spike sorting, single cell isolation. So the spatial information is already used into that. So we have a very single event or a single cluster sitting there. Of course, there are other factors, like I have not talked about that, axon propagation. So sometimes you see the axon propagating through that. So we haven't included that yet. But yes, the spatial information of the spike itself is included in the first step in the spike sorting.